Welcome back to week 2, lecture number 5 of Introductory Animal Physiology. After our first lecture, we hope you are ready to dive deeper into the nervous system. In this lecture, we'll explore action potential, synaptic transmission, neurotransmitters, and sleep. Let's continue building our understanding. Neurons. Neurons' main function is what? Its main function is to communicate. So how it communicates? It communicates one neuron to another neuron through the synapses. Now these synapses is of two types. One is of electrical type, another one is chemical type. Now, can all the cells become neuron? Is it possible? Yes, whichever the cells are possessing the properties of excitability and then the electrical signal propagation. So that cells are called as neurons. Now these neurons also show certain physiological properties. The first and foremost property is that resting membrane potential. What is this resting membrane potential? Resting membrane potential is nothing but the membrane potential of a neuron at the static state or at the rest. So this potential is called as resting membrane potential. Usually the resting membrane potential of a neuron is around minus 65 millivolt, or you can say minus 70 millivolt of many excitable cells. Next important physiological property is that graded potential. So graded potential, you might have heard of the grade cards. That means it is related with some higher marks, lower marks related with that. The same way here the graded potential means the potentials which may be somewhat goes towards the depolarization side or somewhat is moving towards the hyperpolarization side or negative side, or more positive, more negative. Next will be the equilibrium potential. The equilibrium potential is expressed as E ion. For example, if it is sodium ion, it will be written as ENA. So this is an equilibrium potential. So equilibrium potential means this is the membrane potential derived at the equilibrium of an ion. That means when the intracellular fluid and the extracellular fluid, where the electrostatic force as well as the concentration gradient meets at the equal point where there is no further exchange of ions will take place, that potential is called as equilibrium potential. All the ions in the body will try to achieve this equilibrium potential, but body won't allow. The next is that property is that action potential. Action potential is nothing but it is having the overshoot as well as undershoot. That means it's having three phases. One is depolarization, repolarization, and then hyperpolarization, which is involved with the different ion channels opening and closure. Next will be the once the action potential is generated, then it has to be propagated. So these are some of the important properties of a neurons. Then coming to the ionic distributions, there are four important ions in the body, or ion channels you can say. The first is foremost is the sodium, next is the potassium, then calcium, and then chloride ions. Based on the activation and inactivation of these channels, the neurons may be depolarized or it may be hyperpolarized. Then why the resting membrane potential is always expressed in a negative, either it is minus 65 we can say or minus 70 millivolt. Why we always express in terms of negative? It is why it is not positive, though the inside of the cell contains lot of potassium ions which is of positive in nature. This is because of the fixed anions. The concentration of the fixed anions is present at higher amount than the outside. That is why the resting membrane potential is always expressed in terms of negative. These fixed ions are nothing but the proteins, nucleic acids, carboxylic groups, and the metabolic carrying phosphates, and these organic ions are too large, which cannot comes out from the cell. As a result, it is fixed inside the cell, leads to the contributing for the negative charge of the cell. Now, some of the important terms which is related with the action potential. The first is that resting membrane potential, that is nothing but the potential difference across the membrane of the resting neurons. Already I told it is minus 65 millivolt, or in some books you can say it is minus 70 millivolt, both are correct. Next is the graded potential. I told these are grade means it is some which leads to the membrane potential towards positive side. Some membrane potential, graded potential will leads the membrane potential towards the negative side. So based on that, these graded potentials will be called as an excitatory postsynaptic potentials and then the inhibitory postsynaptic potentials. When the graded potential or the potentials leads to the activation or release of the acetylcholine and glutamate, which is a excitatory neurotransmitter that ultimately causes the opening of the sodium channels, leads to the depolarization of the postsynaptic membrane. This type of synapses are called as excitatory synapses, whereas there are some synapses where it releases the GABA and glycine depending on the graded potential signal, where it will go and binds with the chloride channel in the postsynaptic membrane, leads to the inhibitory signals, that means hyperpolarization it will move towards. So this type of synapses are called as inhibitory synapses. Next will be the trigger zone. So where this trigger zone usually presents, so this is a neuron, right, and these neurons will come here, this is the axon and this is the dendrites. Now the dendrites receive the signals. Now here this is the point is called as a axon hillock. Now this axon hillock is called as a trigger zone. So all the graded potentials will be received through the dendrites and then it will get integrated at the axon hillock, where once it satisfies the next is coming, that is the threshold potential. Once it reaches the threshold potential, then immediately the neurons get depolarized. Right? So threshold potential, this is the capability to depolarize the membrane. Usually it is of minus 55 millivolt. So from minus 65 millivolt, when the axon hillock or the trigger zone reaches minus 55 millivolt, then immediately it causes depolarization. That means it causes the opening of the sodium channels. Now coming to the action potential as such it is. The action potential, the first is that graded potential. So from the neurons, the graded potential is received from the dendrites. So once the dendrites receives the signals, the graded potentials, like somewhere it is minus 5 millivolt, somewhere it is minus 2 millivolt, somewhere it is plus 10 millivolt, like the different types of grades of potentials it receives. So once it receives, it's all get integrated here. That means plus and minus will takes place, and then ultimately once it reaches or satisfies the minus 55 millivolt threshold potential, then immediately the neurons get depolarized. So once it depolarizes, that means it will follow all or none phenomena. All or none phenomena means when you take a gun and if you shoot, then the bullet will come out and it will go and hit the object. That means once you apply the trigger, then there is no stoppage for the bullet until and unless it go and hits the target. The same way, when the threshold potential it reaches, then immediately the depolarization begins and it ultimately go and reaches the axon, which causes the opening of the voltage gated sodium channels. Now this sodium channel, why we always discuss about the sodium channels but not discussing about the other channels? Because the sodium ion concentration is higher in the extracellular fluid than the other ions. And moreover, these extracellular fluids is easy to withdraw and to go for analysis. So that is why most of our understandings is given in relation with the sodium ions but not with the other ions. That does not mean that the other ions are not important. All the ions are equally important in the body and having its own special activities. So these sodium channels will have 4 important states. So if you take the channel, it will have 2 gates. One is called as activation gate. This is called as activation gate, and the another one From here, this is called as inactivation gate. So what happens in the resting state? The activation gate opens, and then inactivation gate closes. Whereas in the activated state, the activation gate in the open state, and then the inactivation gate is also open. Then coming to the inactivated state, in that the activation gate is open and then inactivation gate is closed. And then in the de-inactivation gate, then it will go to its original position. That means the activation gate is in open and the inactivation gate is closed. So this is called as de-inactivation gate. So the first one is resting state, then activated state, and then inactivated state, then de-inactivation state. Now, if you see the phases in the action potential curve, it will follows like this. That means what? Here, this is called as threshold potential, which is minus 55 millivolt, and it starts with the resting membrane potential, that is minus 65 millivolt. So when the graded potentials, when more of positive, graded potentials received, which moves the potential from negative side towards the positive side, it will go from minus 65 to minus 55 millivolt, where suddenly it reaches the threshold potential. Once it reaches, immediately it causes the opening of the voltage gated sodium channels. Then when it reaches at around plus 40 millivolt, then what happens? Suddenly it causes the closure of the voltage gated sodium channels and it causes the opening of the voltage gated potassium channels. This phase is called as repolarization. So here it causes the voltage gated potassium channels to open, and this is called as repolarization phase. Now, why in the depolarization phase it is moving towards positive side? Because when the sodium ion started coming inside, it will move from negative side towards the positive side. Then the equilibrium potential of the sodium ion is plus 55 millivolt. So that is why it is trying to reach its equilibrium potential state. But what happens here is that the body won't allow it to reach up plus 55 millivolt. So when it reaches plus 40, immediately it terminates and then it causes the closure of the sodium channels and it causes the opening of the potassium channels. That results into the backward movement of the potential from positive side to the negative side, because from inside the potassium ions will go to outside. So as a result, inside the cell the negativity will rises. So from plus 40 again it will move towards the negative 0, and then it will go towards the equilibrium potential of the potassium ion, which is around minus 80 millivolt. That is the equilibrium potential of the potassium ions. So when it crosses around minus 65 millivolt, that is the resting membrane potential, then it will try to move towards minus 80 millivolt. That means when it reaches around minus 70 or 75 millivolt, then again body will terminates this channel and then it activates the sodium potassium ATPase pump, where the reversal process will take place. So in this reversal process, what happens? Whatever the sodiums entered in the depolarization phase and whatever the potassium is went outside, this all will come back with the help of the sodium potassium ions, with the exchange of two sodium with the potassium ions. Next is that refractory period. Now when this process action potential is going on, there is a part is called as a refractory period. So in the refractory period, what happens? Whatever the stimulus you give, the neurons will not respond to that. So it is a exhaustive phase, you can call it. This refractory period can be called as a exhaustive period of a neurons. So it can be divided into two types. One is absolute refractory period, another one is relative refractory period. So absolute refractory period means even if you provide a very strong stimulus, then also the neurons cannot able to excite or it cannot depolarize. So refractory period is called as absolute refractory period, whereas the relative refractory period, in that if you are going to produce a stimulus which is above than the normal, then it can able to produce another depolarization of the membrane. So this phase is called as relative refractory period. Now coming to the propagation. So far we discussed about what is action potential, what are the different phases are there. Now once this action potential is generated, then it has to be propagated. Otherwise it is of no use. The signal is produced, then it has to propagate. Now how it will propagate? You all have seen the sea or any other water body. In that you might have seen the tides. So before tide comes, the water surface is very plain, plateau. But when the tide comes, then it will give you this type of one. And then if you see the before and after the surface of the tide, it is plain. Whereas in between, you can see a small bump like structure. So the same thing happens in the neurons also. So here what happens, how it propagates? By passive spread of the current. By slowly it is opening the nearby voltage gated sodium channels. That is how it is possible for the tide or for the movement or for the propagation of this action potential depolarization from the axon hillock to the axon terminal is possible. So because of this passive spread of current by opening the voltage gated channels of the nearby membranes. So this is how it causes the action potential propagation. Now this propagation depends on two factors. The first factor is that diameter of the axon, or how large the diameter is, that much easy for the signal to pass to the axon. It is like if the road is wider, then we can move faster. If the road is smaller, then it will take time to reach the destination. So the same way, when the diameter of the axon is wider, that means it is easy for the impulses to reach the target organs very easily. Then the next is the resistance. If you have a very less number of vehicles in your track, then you will take time to reach the destination because of the traffic. Whereas if there is less number of vehicles or there is no vehicles, then it will be easy to reach the destination within short time or less time. So the same way, the resistance play a major role even in the nerve generation as well as the propagations, because it leaks out the current. Then the next important is that myelination process. Of course, in the last lecture itself, I told you about the myelination and the myelin sheath, which cell is responsible and what is the function of this myelin sheath, which acts as an insulator, and then that is results into the saltatory conduction. Saltator means it is leap. Leap means it is movement like this. So next is that synapse. Once it reaches the axon terminal, then the next is that it has to transfer the signal to the next neuron or the next target organ. So then only the signal will be passed or the purpose is solved. So the synapse is nothing but these are all the specialized contact areas where the signals will be mediated from one cell to another cell with a synaptic cleft in the middle. Now here there are two types of synapses. I already told, one is electrical synapse, another one is chemical synapse. So the electrical synapse is where a small gap junctions are present, electrical gap junctions, through that the current passes. So these type of electrical synapses are present in very few areas in the body, such as hypothalamus, hippocampus and thalamus. Whereas the chemical synapses is present widely in the body. So this is mediated through the chemical messengers that is called as the neurotransmitters. Now of course there are different types of neurotransmitters are there that we will see. Now next coming to the synaptic transmission. Now how in this chemical synapses, the transmission is taking place. When the action potential, the depolarization reaches the axon terminal, it causes the opening of the voltage gated calcium channels at the terminal, where the calcium enters and then it binds with the troponin C, where then it causes the synthesized and the stored neurotransmitters in the reserve pool as well as the readily available pool get mobilized, where it get docked, and then once it get docked at the membrane level, then it releases slowly by an exocytosis process where the fusion and release is taking place. Now once it releases in the synaptic cleft, then it will go and binds to the postsynaptic membrane, where it will have the different types of channel that is called as ligand or chemical gated channels. Now so far we discussed about the other type of channels, that is the ligand gated and the chemical gated channels we discussed, right? Whereas in the earlier this one you might have seen the voltage gated channels we discussed, right? So far we discussed voltage gated, but suddenly how come it became a ligand gated? Ligand means it is a chemical. So here what is the chemical messenger? The chemical messenger is a neurotransmitter. So that is why this channels is because of this neurotransmitters, it is opening that channel. So due to that reason, these channels are called as a ligand or chemically gated channels, but not the voltage gated channels because of difference in the potential. Now these channels, either it is ionotropic or metabotropic. Ionotropic means the channels, when it binds, then simply it causes the opening of the ion channels. That's all. When the acetylcholine or the epinephrine or norepinephrine, whichever comes and binds with the postsynaptic membrane, then immediately it causes the opening of the channel. So that channel is called as ligand gated ionotropic channels. Then metabotropic means when it binds at the postsynaptic membrane with the receptor, then immediately it triggers the secondary messenger system inside the membrane, and then that causes the opening of the ion channels. So secondary messenger system activation, either GTP or cyclic AMP or inositol triphosphate or calcium mediated, there are different types of secondary messengers are available in the body. Based on that, the biological response of the neurons takes place. Now once these neurotransmitters are released in the synapse, then what will happen to these neurotransmitters? Once its function is served, then it has to be removed from that place, otherwise it causes a detrimental effect to the neurons as well as the body. So the first and foremost fate of the neurotransmitters is the enzymatic inactivation. So simply, for example, acetylcholine esterase is there. This is an enzyme which will come and destroy the acetylcholine which is present in the synaptic cleft. So that means it cleaves the acetylcholine into the acetyl-CoA and then the choline. So then it will be taken back, and then again it gets synthesized in the presynaptic membranes as a neurotransmitters. Then next is the diffusion mechanism. Once the purpose is over, then immediately these neurotransmitters will be diffused back in the neurons. And there is one more way is that is missed here, that is called as reuptake mechanism. Mostly if you take the astrocytes, the perivascular end feet of the astrocytes, where the reuptake of the glutamate receptors or the glutamate neurotransmitters is done. So these are the three important fate of neurotransmitters. Next, what is that neurotransmitters? Neurotransmitters, I told several times, these are nothing but the endogenous chemical messengers that transmit signals from one cell to another. Now the key neurotransmitters are acetylcholine, glutamate, epinephrine, norepinephrine, GABA and glycine. So already I told that neurotransmitters is of two times channels which will follow. One is ligand gated, another one is voltage gated. Then synaptic transmission, already I discussed. Then acetylcholine, how the enzymatic inactivation is taking place here with the help of the acetylcholine esterase. And then the receptors. Receptors are nothing but the specialized protein which is found on the surface of the neurons, where the other cells that receive and responds to the chemical signals. So ionotropic receptors, I told, it just causes the opening of the ion channels, whereas metabotropic receptors, it will just activate it based on the secondary messenger systems. It will go and causes the opening of the channels. Then There are other types of receptors such as cholinergic receptors. That means these are the receptors which are responsible, which will get activated with the help of the acetylcholine. That is why it is called as cholinergic receptors. Then adrenergic receptors. These are the receptors which get activated by the adrenaline and non-adrenaline, and glutamate receptors, where these receptors bind with the glutamate, then it will get activated. Of course, there are different subtypes are there in these cholinergic, adrenergic and glutamate receptors. Now, the last part of our today's topic is sleep. Better please don't go to sleep, but the topic is only the sleep. Okay, why the sleep is necessary here? We need to sleep properly because it will replenish the brain biochemically and functionally safe, and it is required for consolidation and strengthening of the memories and learning. For this purpose, the sleep is necessary. So that's why you need to have a sound sleep to store your memories and the learning to take place properly, right? So after this class, you can go and sleep. Those sleep states, if you take, there are slow wave sleep or non-rapid eye movement sleep, and then rapid eye movement sleep, or another name is that paradoxical sleep. Now, the process of falling asleep, how, of course you know, but still I will tell. The first is that awaken, right? You will be awake. So during this phase in the EEG, electroencephalography, where the beta wave is produced, then next will be the drowsiness. Yes, little bit drowsy, you will get the sleepy, right? So alpha wave, which is in the EEG, you can say the alpha wave, you can see where it is called as N1 stage. The next is that light sleep. Slowly you got your eyes are getting closer, where you get the theta waves, which is seen in the EEG machine, and then that this is also called as N2 stage. Then the deep sleep, which is a delta wave in the EEG, you can see that is called as N3 stage. Now, the sleep cycle, if you say, when you slept, then first you will go for a light slow wave sleep, then followed by the deep slow wave sleep, then followed by again light slow wave sleep. Now, all these three slow wave sleep, light, deep, light, these are all under the control of the parasympathetic division of the autonomic nervous system. So that means you are under normal, right? Your system is normal. Whereas the next stage is the REM sleep. The rapid eye movement sleep is also called as paradoxical sleep, because in this you are under the control of the sympathetic division of the autonomic nervous system, where you will see the beta wave. Beta wave, I told, when you are awake, then only in the EEG it will get recorded as a beta wave. But here you are sleeping, that too in the rapid eye movement sleep, where in the sympathetic, under the control of sympathetic nervous system, you will see the beta wave. So that is why the REM sleep is also called as paradoxical sleep. Right? You are sleeping, but you are awakened. Whatever the body, when awake, whatever it is seen, the same thing will be seen during the REM wave sleep. Then the cycle length, the sleep cycle length, if you see in different animals, in humans it is 90 minutes, and then in primates it is 60 minutes, dog and cat it is 20 minutes, rats it is 10 minutes. Now, out of this, all around 25% is accounted for the REM sleep, and birds it is 10 seconds. Now, which are all the animals can sleep well? The predator animals are usually sleeps well, and then if the animal is sleeps in their home, or we are used to sleep in our home, that means we sleep having a sound sleep. The same way the animal sleeps in their own den, sleeps more than the prey animals or the shelterless animals, because anytime the predator will come and attack on them. Then there are some animals you can see where one cortical hemisphere is active, another cortical hemisphere is inactive. So this is because as a nature provided it is as a mechanism of a vigilance. So what are all the animals you can see? The dolphins, whales, manatees, seals, ducks, alligator. These are the animals sleeps where one hemisphere is active, another hemisphere is inactive. That means one eye is open, another eye is closed. So today, if we summarize, then sensory stimulus we have seen, then the dendrites, then soma, graded potentials, right? And then we have seen how the synapses get transmitted, synaptic transmission is taken place. Then what are the different types of receptors? The cholinergic receptors, adrenergic receptors, then neurotransmitters we have seen. Then how the neurotransmitters reuptake has taken place, excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Then what are the different types of other types of receptors such as cholinergic, adrenergic and glutamate receptors. Then what is sleep and what are the different states of the sleep. Then what is the process of falling asleep, and what are the different animals in that the different sleep states are there. So in this we completed the second objectives, and I will see you in the next class. Thank you.